Hello everyone, hope you are doing great. Let's start with today's class. And today's class is regarding basal ganglia. Basal ganglia, it is the term given for the collection of the neurons within the nucleus. Like basal ganglia, they are also called as basal nuclei. And these are collection of the neurons within the nucleus, and that is present at the base of the brain. That's why they are called as basal ganglia. So before we proceed further, I presume that neuroanatomic part hasn't been started yet, right? So you might not be aware about embryology of the nervous system. So before proceeding further, let me just give you brief overview regarding the embryology part of the nervous system, so that few of the terms I will be using in the coming slide, you will get familiar with those terms, right? So the if we look in a broader aspect, all the tissues, they can be traced back to three primary germ layer: ectoderm, mesoderm and endoderm, right? And this formation of the three germ layers, ectoderm, mesoderm and endoderm, this process is called as gastrulation, right? Gastrulation. And this gastrulation, it occurs during the third week of the embryogenesis. And this entire process, this formation of the three germ layer, is called as gastrulation, and it occurs or happens during the week 3 of embryogenesis. And it is initiated by the formation of primitive streak, right? This primitive streak. What is this primitive streak? This one. It is thickening of the epiblast cell layer. See, there will be two layers initially, that is the epiblast layer and the hypoblast layer. This pinkish one, hypoblast, and the blue is this sky blue color, this is the epiblast one. Okay. So this gastrulation, it is initiated by formation of the primitive streak, this dark blue color. And what is this primitive streak? It's thickening of the epiblast cell layer that appears at the caudal end of embryo, and it starts, because it is appearing at the caudal end, it will grow cranially. Okay. And epiblast cells, then it will go after the formation of the primitive streak. What will be the effect? It will convert the epiblastic cell to undergo from epithelial to mesenchymal transition. Okay, epithelial to mesenchymal transition. What does it mean? It means it will make them to get converted few of the cells into mesenchyme. And how will it happen? It will happen by these epithelial cells, they will lose their cell to cell adhesion characteristic, and because of the losing of the cell to cell adhesion, it will allow them to migrate downward through the primitive streak, like this black arrow. It is showing the movement of these epithelial, few of the epithelial cell which has lost their adhesion with each other, they will migrate downward through the primitive streak to form mesoderm and endoderm. And the remaining part of the epiblast will form ectoderm, right? See, like this yellow color, it is endoderm, and this endoderm is displacing this pinkish one, the hypoblast, and it will appear like this is the starting stage, 14 to 15 days, like the initial, the first day of the third week, and within 1 to 2 days this change will happen. This is the primitive streak, this is the ectoderm, this in between reddish color, this is the mesoderm, and yellow layer will be, and this one reddish color is mesoderm, sorry, and the yellow one is endoderm. Okay, like after within 2 days of the gastrulation begins, some of, like this is on the 16th day. Now coming to the next part, this, like this is the neural tube formation happening on 19 days, like within 2 to 3 days after the gastrulation begins, What was this blue color? Blue color was ectoderm, this was mesoderm, and this was the endoderm. So, on the second to third day, or we can say on the 19th day, some of the epiblast cell layers, they will migrate cranially through the primitive node. And what is the primitive node? That is the cephalic end of the primitive streak, to form a midline cellular structure that is called as notochord. Like few of the epiblast cells, just remember, few of the epiblast cells, they will form the notochord. Now, what does this notochord do? This notochord will induce the overlying ectoderm to differentiate into neuroectoderm. See, this blue is, and initially this was entire blue, sky blue color. After the formation of the notochord, it will induce the overlying layer of the ectoderm to get converted into other form, and that other form is means the ectoderm is getting differentiated into neuroectoderm. This purple color is neuroectoderm, right? And what will happen? It will induce it to get converted into the neuroectoderm, and these neuroectoderm will move toward the midline, like this arrow and this arrow. This peripheral end from left side and the right side, they will try to move medially, okay, medially, and this will give rise to formation of the, this is called as neural fold, okay, neural fold. How the neural fold is formed? Neural fold is formed by the movement of the peripheral end, both peripheral and left and right, toward the midline, okay, and once they reaches in the midline, they will form a tube that is called as neural tube, okay, and in between this purple color and this ectoderm, the in between layer, the greenish layer, these cells, they will migrate at the underlying of this neural fold, and they are called as neural crest. So, this will form the neural tube, this will be neural crest, and this part is notochord, these three things, right. So, in detail, if we go over here, like say this one, on the 19th day neural groove is formed. This, like the central line is called as neural groove. This, like, no, this part, this is called as the neural groove, and neural plate will form the neural tube, and when it forms neural tube on the 20th day, in between layers, the blue and the purple, the greenish one, they will form the neural crest. So, on the 21st or the 22nd day, there will be the formation of neural tube, and remaining part of the ectoderm will cover the entire thing, and this is the surface ectoderm. Now, this reddish one is the mesoderm, this one is the endoderm, this part is the notochord, and this is the neural tube, and these greenish one, they are the neural crest. What is notochord's derivative? Notochord derivative is nucleus pulposus, right? Nucleus pulposus. It will become that nucleus pulposus of intervertebral disc. Now, we can say, like if we look at this part, the nervous system, we can divide, it is made up of three primary vesicles, the red one, blue one, and the green one. The red one is forebrain or the prosencephalon, and blue one is midbrain, the mesencephalon, and this one is the hindbrain or rhombencephalon. So, three primary vesicles, progen, meson, and rhombencephalon. They will give rise to 5 secondary vesicles. Prosencephalon will form 2 secondary vesicles, telencephalon and diencephalon. Mesencephalon, the primary vesicle, will stay as single only, the mesencephalon secondary vesicle. And this rhombencephalon, what it will become? It will become two things, metencephalon and myelencephalon. So, we can say there are three primary vesicles, progen, meson, and rhomben. which gives rise to 5 secondary: telan, dian, mesan, metan and myelencephalon. Now, if we look at this one, the telencephalon gives rise to cerebral hemisphere and basal ganglia. This will give rise to cerebral cortex and basal ganglia, and the cavity within it will be the lateral ventricle. So, telencephalon forms lateral ventricle, cerebral cortex and basal ganglia. What will diencephalon do? Diencephalon will form thalamus and hypothalamus, and like say these are the lateral ventricle. I am just presuming this part is the lateral ventricle, left side, right side, and it gives rise to the third ventricle. So, third ventricle is formed from the diencephalon. Mesencephalon will form the midbrain, and what is lying within the midbrain, the counterpart of this ventricular system, the cerebral aqueduct, right, the cerebral aqueduct. Now, the rhombencephalon is forming metan and myelencephalon. Metencephalon gives rise to, or the adult derivative of the metencephalon is pons and cerebellum. And what about the ventricular system? Upper part of the fourth ventricle. And myelencephalon forms medulla and lower part of the fourth ventricle. Now, the sequence, it is easy to remember the sequence. Dian is coming at the second number, second number. Okay. Telan is, you can remember like telomere or teler. Tel means the end part. So, the upper end is telencephalon. The number 2 is diencephalon, that is why the di, and afterwards there are 3 M, and all these 3 M's are arranged as alphabetically, like S, then T and then Y, mesan, metan and myelan. Okay. So, that is the like embryology part of the nervous system. The second thing we need to know before going into detail regarding the basal ganglia is this one you have already read in your 11th 12th standard, but still we will just quickly go through it. What is this line? This is the central gyrus, so the central sulcus of the Rolando. It is not Ronaldo, it is Rolando, right? And the post central gyrus, the posterior part, or this one, this is the anterior part of the central sulcus and this is the posterior part. So, the post central gyrus forms primary somatosensory cortex, area number 3, 1 and 2, right? And the precentral is primary motor cortex, area number 4, this yellow one. And anterior to this primary motor cortex, yellow one, this part is premotor. This is the motor and this one is premotor area. And anterior to the premotor area is this frontal cortex area. This is called as frontal eye field, and it helps in controlling the voluntary contralateral horizontal gaze of the eye movement, right? And post central gyrus, this blue one was primary somatosensory cortex, and this green one is association area of the somatosensory. This yellow one is visual association area, this occipital gyrus, and this is the primary visual cortex. Okay. So, I just wanted for you to remember these 3 things, like we will be using these terms very frequently: the premotor, primary motor association, and the somatosensory as well as the somatosensory association area. So, you must be aware about where exactly it is located, like the primary motor is precentral and anterior to the primary motor is the premotor, and post central gyrus is the somatosensory, and posterior to that is somatosensory association area. Okay. Now, the regular diagram. See, there are few things in the nervous system, like few of the diagrams or the flow charts. There is 1 or 2 more. Out of those, like flow charts or the diagram, you can easily squeeze in anything related. If examiner asks you any question, you can squeeze them in easily. Like say, if there is a question regarding the motor, anything related to the motor, like the motor tracks, like the descending tracks, or the corticospinal tracks or corticobulbar tracks. Then again, like in the coming classes, we will read about these descending tracks. They are broadly divided into two parts, the pyramidal tract and the extrapyramidal tract. The pyramidal is because they are forming the pyramid. The corticospinal, corticobulbar, they are called as the pyramidal. And the other descending tract, that is mainly coming from the basal ganglia. For the time being, just remember, mainly coming from the basal ganglia, they are extrapyramidal tract. So, if there is any question regarding the whether pyramidal tract or the extrapyramidal tract or broadly like the descending tract or anything related to anything to do with the motor system, like the maintenance of the posture or the movement, or to the basal ganglia or to the cerebellum, or the control of the movement via the upper motor neuron, just squeeze in this flow chart. Okay, this squeeze in this flow chart. So, the first and foremost thing for you is to mug up this flow chart. It will be very helpful. So, what was it? It was like whenever we get any idea of any voluntary movement, these three champions, the basal ganglia, the lateral part of the cerebellum and cortical association area, they form a huddle and do the talking, means they do the planning, how we are supposed to perform or execute this idea. And after that, they reach to consensus. They will ask the boss, the cortex, the cortical association area, the basal ganglia and the lateral cerebellar hemisphere. They will ask this boss, the premotor and the motor cortex, to send the order to the lower motor neurons within the spinal cord to perform the movement, right. And one of the copy of that blueprint is sent to the cerebellum. Which part? Intermediate. Lateral part of the cerebellum do the planning and intermediate part of the cerebellum do the execution process. Okay. And this premotor and motor area send one copy to the intermediate cerebellum, and whatever the movement is taking place, one copy of that movement comes back to the cerebellum. So, cerebellum has got two copy, one which is supposed to be performed, the other copy which is in actual getting performed. So, accordingly it will balance the thing. It might automatically can directly bring the changes, any like refinement of the movement, or it will send back its feedback to the premotor and this one premotor and motor cortex area. And whatever the movement is getting performed, it will also send a copy to the premotor and the motor cortex. So, this is kind of an interwoven circuit, how any idea gets planned and it's executed in the form of the movement. So, where exactly is today's topic basal ganglia? It is within the planning and programming of the movement. It does the planning and programming of the movement, and through that it has got effect on, we can say the basal ganglia has got the effect in this execution of the movement, planning and execution of the movement in the form of cognitive process. Cognitive means to think about, thinking, control, okay, and as well as in performing, that we will cover in the coming slides. Okay. So, we can say broadly, this is one more flow chart we can say. The basal ganglia and the cerebellum, they modify the movement on a minute to minute basis. Motor cortex sends information to both. The output of the cerebellum to the cortex is excitatory, while the basal ganglia are inhibitory. See, this motor cortex, it is sending the blueprint to the motor neuron, interneuron, which is present within the spinal cord, and the movement is getting performed, right. And from the peripheral nervous system or this skeletal system, the muscle which are performing the movement, one copy is coming back to the cerebellum, other copy is coming back to the basal ganglia, right. And what I said, the cortex is sending a copy to the cerebellum via pons. Okay. And cerebellum is again interacting with the cerebral cortex via thalamus, and basal ganglia is contacting the cerebral cortex via thalamus, because the gateway of the cerebral cortex is thalamus. So, by this flow chart you can easily see that one is excitatory and other one is inhibitory. So, the connection of the basal ganglia to the cerebral cortex is, we can say, it is inhibitory, and the effect of the cerebellum to the cerebral cortex is excitatory. What does it mean? Means whatever the planning and programming of the movement has been done by these three parts, the cortex, basal ganglia and cerebellum. Now, it is turn for the cortex to execute that movement, and as this line says, the basal ganglia and cerebellum, they modify the movement on minute to minute basis. So, this modification which is happening with the help of the cerebellum, main concern of the cerebellum is, like if there is sluggish type of the movement, it will excite it, means remove the sluggish part, and main control or the main function of the basal ganglia for this minute to minute modification of the movement is, it will try to inhibit it. That is the broader aspect of saying. But again, when we go into the detail about the basal ganglia, like see, anything what is happening around us or within us, it has to be balanced, like the inhibitory as well as excitatory part, it has to be in the balance. If anything happens more, inhibitory is more or the excitatory is more, there will be the problem. Okay. So, we can say the basal ganglia mainly it is sending the inhibitory, but within the basal ganglia, like how much inhibition it is supposed to send to the cerebral cortex, it should be balanced. Okay. So, what is the difference between the basal ganglia and cerebellar motor loop? Like the connection of the cerebellum with the cerebral cortex and connection of the basal ganglia with the cerebral cortex. Basal ganglia receive input from the most of the areas of the cerebral cortex, whereas input to the cerebellum is from the cerebral cortex is very restricted. What does it mean? It means the basal ganglia is receiving the signals or the connections from the cerebral cortex from the premotor area, the primary motor area as well as the association areas, but cerebellum is receiving only that blueprint of the movements that is going to happen, and that blueprint is final part. Like who plays the final part in making the blueprint? The motor cortex. So, mainly the motor cortex is giving the output to the cerebellum, but to the basal ganglia from the motor as well as the premotor as well as association areas. That is the one thing. Now, the output from the basal ganglia is not only going to premotor motor as it is going from the cerebellar, but it also goes to the prefrontal cortex. It goes to the prefrontal cortex too. And what was the prefrontal? Where exactly the prefrontal area? Like precentral gyrus, the initial part was the motor, anterior to the motor was premotor, and anterior to the premotor is prefrontal. So, basal ganglia output goes to all these three areas, but cerebellum has got connection, it has got to do with motor on the premotor. Motor gives connection, like sends the signal to the cerebellum, and cerebellum sends back to the motor and premotor. But basal ganglia input as well as output connected with the cerebral cortex is widespread. Basal ganglia do not receive somatosensory information from the ascending tract through the cerebellum, though the cerebellum do get. The third part is, as we know, the blueprint is sent to the cerebellum as well as to the lower motor neuron to perform the movement, and cerebellum does the matching, means it has to get a copy back from the ascending tracks, like what kind of the movement is getting performed, but basal ganglia do not get. So, these are the three difference between the basal ganglia and cerebellum. First is input to the cerebellum and output from the cerebellum in connection with cerebral cortex, it is limited, it is concerned with only motor and premotor area. but both output and input of the basal ganglia to the cerebral cortex is widespread. It involves so many areas, not only the motor and the premotor, it also involves the prefrontal association areas, right. The third is the somatosensory, what kind of the movement is being performed will be received directly to the cerebellum, but it will not go directly to the basal ganglia. Okay. So, this is one more connection like the first diagram I said to squeeze in, that is the another aspect of showing like the motor cortex, basal ganglia, cerebellum, these three they do the planning, then this blue cortex spinal tract goes to spinal cord and muscle movement is performed, and somatosensory ascending goes to the cerebellum, but it does not go to the basal ganglia, right. Now, coming to the main topic, that is the basal ganglia. So, what exactly is the basal ganglia?